Good afternoon, everyone. This is Group 10 with Infinity Woodruff, Nathaniel Guff, Harrison Stuckey, and myself, Daisy Velassi. Today we'll be presenting our research on the effects of pollution on terrestrial plant species. Our study examines how pollution impacts plants at both the phenotypic and genotypic levels, with a focus on terrestrial ecosystems. By understanding how plants respond to pollution, we'll be able to contribute to conservation efforts and predict long-term environmental impacts. Thank you. Before we get started, here is a quick overview of what we'll be covering today. We'll start with background information, outline why this research is important, then we'll present our research question and methodology, detailing our approach to the data collection and analysis. Next, we'll discuss our timeline and expected outcomes. Finally, we'll wrap everything up with the significance of our research, followed by our conclusion and references. To dive a little bit deeper, pollution is a major environmental stressor affecting plant ecosystems. Industrial activities, urbanization, and agriculture release harmful pollutants such as heavy metals, phosphates, and volatile organic compounds. These contaminants impact plant biology in two key ways. First, phenotypically, by altering physical traits such as stomatal density, root structure, and pigment composition. And secondly, genotypic, by triggering differential gene expression and mutations that influence plant adaptation. Our research aims to explore how pollution impacts plants differently in terrestrial environments, addressing key gaps in current scientific understanding. To go even further, we will now dive into the background. Plants respond to pollution in distinct ways depending on their environment. Terrestrial plants are exposed directly to airborne pollutants and contaminated soils, leading to stress-induced changes in morphology and gene expression. A key gap in existing research is the comparative analysis of pollution resistance across different plant species. Some invasive species may be more tolerant to pollution, while native species could be more vulnerable. Because of this, by investigating these differences, we're hoping to provide insights that support conservation efforts, phytoremediation strategies, and agriculture sustainability.
Now that we have a little bit of background information, we are going to discuss the project at question. What we want to know is how has pollution on terrestrial environments affected plants both genotypically and phenotypically? What we'll be exploring is how pollution alters plants, both phenotypically and genotypically. This is observable traits, phenotypically, and gene expression and mutations, genotypically, and how this affects across terrestrial ecosystems. By understanding these plant responses to pollution, this can help inform conservation strategies and predict ecosystem resilience.
Hi, my name is Infinity, and I will be discussing the methodology for Group 10's capstone project. So, in a more traditional master's program, you would actually be starting with doing some field work, collecting samples for your research, doing extraction of DNA, RNA, whatever your interest is. Then doing library preparation, sequencing, and then ending with the analysis of all of that data. But for this program, we will be starting from basically step 4 in this picture with the analysis using open source data that is available online. So let's speak about that. So I will be responsible for the NGS analysis part of our course project. The repository that I will be using is called Bioconductor, and this is actually going to be in RStudio. This open repository has tons of different tools that are great for NGS analysis. I will be acquiring the skills to be able to use these tools within Bioconductor through DataCamp. There is a skills track called Analyzing Genomic Data in R. So I will be going through that, following all the lectures there, and then practicing on my own with data that we gather. And in addition, I will also be going to YouTube and watching tutorials of other people using the tools within Bioconductor because I find that there are tons of different videos online that you can actually watch somebody using different types of data with these tools. So some of the main packages within Bioconductor that I will be focusing on are BioStrings, Genomic Ranges, DSeq2, RBowtie2, and GViz. So BioStrings is good for the manipulation of large biological strings. This would be good for annotation and aggregation of data. Genomic ranges is good for finding target regions of the genome. This also would be good for annotation and aggregation of data. Third is DSeq2. This is good for identifying genes and pathways along with determining read counts. And this would help us with confidence and variability between species. And lastly, I would be focusing on RBowtie2 and GViz, and this would be used to align data to a reference genome and plotting it. And this would be good to help us identify different types of mutations. So these are just a few of the packages within Bioconductor that I will mainly be focusing on, but if I find that there are more in Bioconductor that would help us answer our project question, then of course I would be adding that because there is just so much to choose from within Bioconductor.
In order to analyze how pollution affects plants, we will first need to collect comprehensive environmental and biological data. By doing this, our group has decided to look into site selection and categorization. We're going to be doing this through selected data that is already accessible online. We're going to collect data from plant samples from industrial, agriculture, and urban sites with varying pollution levels. We'll also be looking at soil samples that will be analyzed for pH, heavy metals, and nutrient levels to measure pollution intensity. From there, we'll look at plant sampling and trait analysis. This will be used to measure stomatal density, root structure, and pigment composition to assess observable adaptations to pollution. We'll be focusing on comparing native and invasive species in order to determine which plants show a greater resilience. We'll also be looking at molecular analysis, where RNA will be extracted from plant tissues to examine gene expression, changes due to pollution exposure, and quality control measures like a bioanalyzer and fluorometry, which will ensure high-quality genetic data. By integrating molecular data, we will correlate environmental pollution levels with plant stress responses, providing valuable insights into adaptation mechanisms. I will also be focusing on data manipulation and pre-processing within Python. Once our data is collected, we will need to process, clean, and analyze it efficiently, and that's where Python is going to play a crucial role. First, we'll do pre-processing environmental and sequencing data. I will use pandas and NumPy to structure data sets, handle missing values, and normalize pollutant concentrations across sites. Next, I'll be looking into genomic data processing, where BioPython will be used to filter low-quality reads, trim sequences, and prepare data for differential gene expression analysis. And then lastly, I'll be looking into data visualization and interpretation. Using SciPy and Matplotlib, we will generate statistical distributions and visualizations of pollutant gradients. This will allow us to correlate pollution levels with plant stress responses, reinforcing the connection between environmental stressors and plant adaptation. By using Python, we will ensure efficient data handling, enabling us to extract meaningful insights that contribute to our overall research objectives.
Another method for exploring the research of pollution and its effects on plants, both land and aquatic, is using linear regression. By using linear regression, it can help quantify these relationships between pollution factors and plant traits. Pollution factors are the independent variable. So something such as lead would be lead within the soil levels. That would be the independent variable. And what's being the dependent variable is the plant trait. Something such as like gene expression levels or whatever it may be. The model down here is just showing an example of how it's used in R. And essentially, this linear regression will help us identify whether pollution significantly affects the traits or if it does not. These skills can be acquired through using DataCamp and this statistical fundamentals in R, as well as looking at some old notes from the previous classes in LSC 540 and LSC 541. This presentation had not been updated, but it has been edited to say LSC 541.
Hello, my name is Harrison Stuckey, and I will be going over the last of the methodology section and the timeline for our project. In addition to the NGS analysis and linear regression, we will also apply statistical genetic methods in the form of genome-wide association studies, or GWAS, to identify genetic variants associated with pollution tolerance. The GWAS methodology can be broken down into 4 parts. First, we will obtain genotypic data from plant species exposed to varying levels of pollution. This data will be sourced from public repositories or generated from the next generation sequencing dataset. In addition, phenotypic traits such as growth rate, leaf area, root structure, stomatal density, and pigment composition will be measured or extracted from databases like the Tri-Plant Trait database. Once the datasets have been created, we will filter and normalize the data in applications such as R or Python and export them to be used in specialized GWAS software called Tassel. And with Tassel, we will perform association analysis to identify genetic markers significantly correlated with phenotypic traits under pollution and stress. The software employs statistical models to account for population structure and relatedness, reducing false positives and ensuring robust results. Lastly, we can export the results and data to be visualized and compare with our gene expression analysis. Ultimately, GWAS complements linear regression and NGS analysis in that we can achieve a more holistic understanding of how pollution affects plants. Linear regression provides a high-level view of the environmental impact, while GWAS delves into the genetic mechanisms driving these changes. Together, these methods enable us to identify key genetic markers for pollution tolerance, predict how different plant species or populations will respond to pollution, and develop targeted strategies for breeding pollution-resistant crops or conserving vulnerable species. So, as briefly mentioned in the previous slide, the GWAS software that will be used is called Tassel, which stands for Trait Analysis by Association, Evolution and Linkage. We chose Tassel in that it has a user-friendly interface, scalability for large datasets, provides comprehensive analysis, and can easily be integrated with other tools such as R or Python for linear regression and data preprocessing. Tassel accepts various file formats, including VCF, or variant call format for SNP data, and HapMap for genotype data. And for phenotypic data, we can import into Tassel easily in a tabular format, where each row represents a plant sample and each column represents a specific trait. The core functionality of Tassel will be the use of statistical models for GWAS. Tassel supports multiple statistical models, including GLMs, or general linear models, which is a basic model for testing associations between SNPs and traits, and MLMs, or mixed linear models, which is a more advanced model that accounts for population structure and relatedness, ultimately reducing false positives. Additionally, Tassel calculates p-values for each SNP trait association and applies significance thresholds, such as Bonferroni correction, to identify statistically significant loci. Lastly, once significant SNPs are identified, Tassel can be used to annotate these loci with functional information, such as gene names, pathways, and biological roles, and we can then export our findings for further visualization or analysis. Here in the slide, we can see an example of what the software looks like, and it displays a linkage disequilibrium plot. We plan to acquire these skills by practicing with Tassel software and then watching any tutorials and reading textbooks alongside with completing statistics modules in DataCamp. Ultimately, our primary focus is a comprehensive differential expression analysis, but we hope to be able to implement Tassel to further enhance our insights. Next, I will briefly go over our timeline for our project. We currently expect this project to be completed in about 13 weeks' time, where the data collection and processing section take up the majority of the first half. First will be literature review and data collection, where we will review existing studies and gather data from various databases. Next, in weeks 6 to 8, after we have identified a suitable data source, we will begin to clean and pre-process our datasets for our experiment. In weeks 9 to 10, we will perform our NGS analysis by performing differential expression analysis in Bioconductor. This will be followed by a phenotypic trait analysis to correlate phenotypic traits with our NGS analysis in week 11. In week 12, GWAS will be conducted to complement our other analyses, if time permits, and lastly, in week 13, we will integrate our datasets to create visualizations of our results to finalize our study. And that concludes our timeline and work plan.
So I will be responsible for the NGS analysis part of our course project. The repository that I will be using is called Bioconductor, and this is actually going to be in RStudio. This open repository has tons of different tools that are great for NGS analysis. I will be acquiring the skills to be able to use these tools within Bioconductor through DataCamp. There is a skills track called Analyzing Genomic Data in R, so I will be going through that, following all the lectures there, and then practicing on my own with data that we gather. And in addition, I will also be going to YouTube and watching tutorials of other people using the tools within Bioconductor, because I find that there are tons of different videos online that you can actually watch somebody using different types of data with these tools. So some of the main packages within Bioconductor that I will be focusing on are BioStrings, Genomic Ranges, DSeq2, RBowtie2, and GViz. So BioStrings is good for the manipulation of large biological strings. This would be good for annotation and aggregation of data. Genomic Ranges is good for finding target regions of the genome. This also would be good for annotation and aggregation of data. Third is DSeq2. This is good for identifying genes and pathways along with determining read counts. And this would help us with confidence and variability between species. And lastly, I would be focusing on RBowtie2 and GViz, and this would be used to align data to a reference genome and plotting it. And this would be good to help us identify different types of mutations. So these are just a few of the packages within Bioconductor that I will mainly be focusing on, but if I find that there are more in Bioconductor that would help us answer our project question, then of course I would be adding that, because there is just so much to choose from within Bioconductor. There are many different reasons why this project would be extremely important, with one of the biggest reasons being conservation efforts. As of right now, there are conservation efforts in place to be able to try and stop pollution or at least slow it down. But as of right now, pollution on land is way far outpacing these conservation efforts that are in place. Some of the major contributors are urbanization, mining, industry, and agriculture. And if you think about it, this pretty much encompasses the majority of jobs on planet Earth. So these contributors are very, very large. The hazardous compounds that are released from these contributors are damaging fragile ecosystems and even wiping out species. And so it's very important that conservation efforts are, better equipped and have more knowledge on how to, kind of just do better efforts. Because if we're damaging our ecosystems and even wiping out species, this is going to be like super detrimental to, the way of life in the future. So I think that this project would be very important for investigating toleration and resistance to pollutants in order to protect ecosystem balances. And even one super interesting thing that I found while doing research on pollutants are that synthetic compounds are stored in fat tissue of organisms that are higher within the food chain. And if you think about this, the highest within the food chain would be humans. So, this topic is actually very important to human health. And these synthetic compounds result in exaggerated negative effects of toxins, especially from plastics that, have hazardous compounds that are broken down into, soil and water and then ends up within our food. And then, of course, it would end up in our body. So it is very important that we not only try and do better work when it comes to saving, plant species and ecosystems, but also we need to keep in mind that it's also going to affect humans in the long run. So I have two graphics here on this slide that I think are, very good at pointing out pollution effects. So here on the left with the tree, you see that pollutants on plants can affect tree productivity. So this would affect our food supply, like say there are, fruit trees and things like that that, provide a food source to humans. If those trees end up not producing their fruit anymore, then this ends up having a negative impact on the food supply for humans. It also can have an effect on plant height growth, leaves falling, and roots weakening. So if this happens, kind of what do you expect for the plant to do? It's not going to be able to really survive long term. And, this is when species begin to be wiped out, and then it's a very big kind of a domino effect within the ecosystem. Here on the right, it kind of shows different efforts that, are phytoremediation. The conservation efforts that are out there for plants definitely have a big emphasis on phytoremediation, and there are many different types of it. So a couple examples are phytodegradation, which is when pollutants are degraded by enzyme activity. And there's also, let me see, phytostimulation, which is when pollutants are degraded by growing roots or microorganisms. So a lot of plants have, a good relationship with different type of bacterias and things like that that could end up, breaking down plastics and things like that within the soil and actually be a good thing. There's also phytoextraction, which is when pollutants are accumulated in harvestable parts of the plant. So, the plant may produce some type of flower or fruit or something like that that, contains that pollutant and would be able to be removed from the environment more easily. And lastly, a big one is phytostabilization, which is when pollutants are immobilized in the soil. So it's not able to make it, like into a plant that is actually a food source and be able to be passed on more down the food chain, like say to humans. So all in all, there are a lot of different things why this project would be very important. So not only for kind of saving and managing ecosystems and also trying to do more research into the toleration and resistance to pollutants, but also is going to end up having an effect on human health in the future. So it is definitely something that is of major importance and more efforts and research is needing to be done to be able to do better in the future.
To conclude, as we've explained before, we expect that pollution heavily alters plant morphology, physiology, and genetic expression. There's a suspectability that invasive species are going to be more resilient to native species based on the hardiness and ability to reproduce and thrive within various environments. Regression models can help quantify the relationship between pollution and plant adaptation, as well as the other methods that were described. This matters because this supports conservation and land use policies, and if we understand this more, it can help us understand biomarkers for pollution and stress in plants and help provide ways and areas for us to improve on in order to help keep the sustainability of the environment.
I just want to go ahead and thank everyone for listening one last time to our presentation on the effects of pollution within terrestrial environments and specific plant species, and how it is all intertwined. These last two slides are just going to be our references and where we got our information from. Thank you again.